grade 8 students, and welcome back to the biology class. We are going to explain each one of these objectives during this effort. At the beginning, let us recall together the main information mentioned in the previous video. We state that blood groups exist in 4 types, which they are blood group A, blood group B, blood group AB, and blood group O. And in order to identify the type of a blood group, we must look for the agglutinogens that are present on the membrane of red blood cell. Remember, agglutinogen means the antigen that exists on the surface of red blood cell, and they exist in 2 types only: agglutinogen A with a circular shape and agglutinogen B with a triangular shape. Also, we state that there is a specific antibodies related to the blood group and exist in the plasma, and the scientific term for these antibodies is agglutinin, which also exists in 2 types, that are agglutinin A and agglutinin B. Both agglutinogens and agglutinin play a very important role in a blood transfusion. Recall again, blood transfusion means the transference of a blood from one person to another. Thus, each type of a blood group is characterized by its agglutinogen and agglutinin. Concerning type A, it represents agglutinogen A on the surface of red blood cell and anti-B in the plasma. However, type B represents agglutinogen B on the surface of the red blood cell membrane and anti-A in the plasma. Now, concerning type AB, it represents both agglutinogens A and B on the surface of the red blood cell where there is no antibodies in the plasma. In contrary, type O represents no agglutinogen on the surface of the red blood cell. It will be empty from any antigens. However, the 2 agglutinins, anti-A and anti-B, exist in the plasma. As we are observing, agglutinogen and agglutinin are opposite to each other in the same type of a blood group. We recall again, concerning type A, there is agglutinogen A, but it must contain in the plasma anti-B. Of course, you are asking yourself now, why the agglutinogen and agglutinin must be opposite to each other in the person's blood? To answer this question, remember please that we stated before, each antibody binds to its complementary antigen. Recall again, complementary means the similarity in shape between the antibody and the antigen. Once complementarity occurs between the shape of the sites of antibody and the shape of antigen, causing them to bind and fit to each other. Similar occur in the blood. Example, concerning this agglutinogen A to exist on the surface of red blood cell and anti-A. If you observe carefully the site of agglutinin A, you observe that it corresponds to have circular site. Mean the circular site of agglutinin A and the shape of agglutinogen A are complement to each other. Thus, they will able to bind and fit together. But what happens when this binding occurs between agglutinogen and agglutinin? Let us see. Once antibody and antigen bind together due to the complementarity in shape between them, this will cause for blood clotting. Blood clotting is the process by which red blood cells clamp together, and its scientific term is agglutination. To understand more the term agglutination and why it is very harmful for our body, concentrate please in the following slide. Suppose that you are of a blood group A, mean you represent in your surface of red blood cell agglutinogen A, and in your plasma exists the antibody A. Thus, there is complementarity in the shape of the site of antibody and the shape of agglutinogen A, causing them to bind and fit together. Once the binding occur now, like represented here in this figure, this is cause for agglutination. And once agglutination exists, it will cause for the death of the individual. Now, what is the relation between all what we mentioned till now and the blood transfusion? Of course, you are still remembering that we stated before at a long time ago, many people died after blood transfusion, and the cause for this death was not clear until the scientist Carl Landsteiner made his discoveries about blood groups, and from that time, blood transfusion became safe. According to the discoveries of a blood group, we state that, example, a person with the blood group A, we recall again, exists on the surface of the red blood cell, agglutinogen A, and in the plasma, anti-B. This individual cannot receive blood from blood group B. However, he can survive in case he receives blood from a blood group O or A. But what is the reason behind the failure or the success of a blood transfusion? Why the person of a blood group A able to receive blood from a blood group O or A, but he cannot do that with a blood group B. In fact, whenever we talk about a blood transfusion, we concentrate on the agglutinogen, mean the antigens exist on the surface of the red blood cell of the donor, and the agglutinin, mean the antibodies in the plasma of the recipient, and that in order to prevent agglutination, mean there must not be complementarity between the agglutinogen of the donor and the agglutinin of the recipient. Of course, you still remember, once we say donor, mean the person who donates, mean gives the blood samples, while recipient is the person who receives the blood samples, mean the patient. To understand well the way we study blood transfusion, concentrate please in this example. Here we have a patient of blood group B, and there are 4 different samples of blood. Sample of blood A, the second one B, the third one AB, and the last one O. What do you think? Which blood sample is suitable for this patient? As we mentioned at the beginning, we must write the agglutinin of the recipient, then we start writing the agglutinogen for each donor. So back to this patient, it's of a blood group B, mean agglutinin A exists in the plasma. Now let us see the agglutinogen for each donor. The first one with the blood group A, mean agglutinogen A, present on the surface of red blood cell. Second one, B, mean agglutinogen B, exists on the surface of red blood cell. Third one, AB, thus the 2 agglutinogens A and B present on the surface of red blood cell. And the last one, O, there is no agglutinogen to be present on the surface of red blood cell. Remember please, whenever there is complementarity between the agglutinin and agglutinogen, it will cause for agglutination. Thus, we talk about the failure of a blood transfusion. So simply now, if we look again to agglutinogen A with a circular shape, and the site of agglutinin A also with a circular shape. Thus, this agglutinogen is able to fit, mean bind with agglutinin A, causing for what? Great, for agglutination. So, of course, the patient will not receive blood from a blood group A. Concerning now agglutinogen B and blood group B, it's with a triangular shape, and that of agglutinin with a circular shape site. Thus, there is no complementarity between the two, mean no binding to occur between this agglutinogen and agglutinin, Of course, now we say the patient is able to receive blood from blood group B. Let us now move to blood group AB, where the two types of agglutinogen exist, agglutinogen A and agglutinogen B. Due to the presence of agglutinogen A with a circular shape that it's complement with the size of this antibody, it will cause them to bind and fit together, mean agglutination take place. Thus, the patient is not able to receive blood from blood group AB. Now, with blood group O, there is no agglutinogen to exist on the surface of the red blood cell, mean there is nothing to bind or fit with agglutinin A. Thus, this type of blood group is able to be given for the patient with blood group B. As a conclusion, person with blood group B able to receive blood from blood group B or blood group O. Once we talk about the failure of a blood transfusion, mean a specific immune response is involved. What do you think? Which type of immune response is involved in the failure of a blood transfusion? To answer this question, remember that there are two types of immune response. The first one, cell-mediated specific immune response, and the second one, humoral immune response. Once we state the term cell-mediated, mean we are talking about the activation of T killer cells. However, humoral, it's relative to the antibodies exist in the plasma. And whenever we want to identify which of these two immune response is involved, we must look for the effector cell that is activated. Back to our question about which immune response involved in the failure of a blood transfusion, remember that whenever there is agglutination, we talk about the failure of a blood transfusion. And agglutination occurs when there is complementarity in the shape between the antibody and the shape of the antigens, causing them to bind together. Since we are talking about the action of antibody, mean the type of immune response involved is humoral specific immune response. Please now take a note in order not to forget that whenever we talk about blood transfusion, we focus on the agglutinogen, mean the antigen of the donor, and not on his agglutinin, simply because we are taking the blood from the donor without the plasma, mean there is no antibodies are transferred. As an application now, please open your booklet worksheets to page 30 worksheet 9 and let us solve question number 10. In this question, they are asking if a person of a blood group A can receive a blood from person of a blood group B, and we must justify the answer. Please concentrate. Whenever we have like this question, it is very important to write at the beginning the agglutinin of the patient, mean the recipient, and the agglutinogen of the donor. And then you make a comparison between the two to indicate if there is agglutination or no. So back to this question, the patient, mean the recipient, is of blood group A, mean in the plasma exists agglutinin B, and the donor is of blood group B, mean agglutinogen B. Let us now look for agglutinin B and agglutinogen B. They are complement to each other, causing them to bind together, and thus agglutination occur. Since agglutination occur, so simply we say that this patient cannot receive a blood sample from this donor blood where the blood is not compatible. Let us together now discuss how we are able to identify blood group. Here exists a sample of blood relative to a person with unknown blood group. To identify the type of a blood group, we must look for the antigens, mean the agglutinogen, present on the surface of red blood cell, which they are antigen A and antigen B. In addition, we must also use the antibodies to identify these antigens, which they are anti-A and also anti-B. So what we are going to do, we are going to put a certain drops of anti-A and also in another one and a certain drops of anti-B. Let us draw two circles, the first one relative to anti-A and the second one anti-B. So we are going to add a simple drop of anti-A for the first one and anti-B for the second one. After that, we will add 1 to 2 drops of blood in each circle and mix well. A few times later, we are going to obtain an anti-A only agglutination occur. Thus, the blood group is A. Why A? Since agglutination occur, mean there is complementarity between anti-A and agglutinogen A. And whenever agglutinogen A exists on the surface of red blood cell, thus mean that the blood group is blood group A. Let us practice how we can identify the blood group in the following two cases. In the first case, where the same type of red blood cells present in the two corresponding samples. For the first one, we add anti-A and the second one anti-B. Let us observe first with anti-A. It represents what? Great, agglutination. Also, if I look for the second one, we observe that agglutination exists. Due to the presence of agglutination in the two with anti-A and anti-B, this means that this red blood cells exist on its membrane agglutinogen A and agglutinogen B. Thus, the type of a blood group is AB. Concerning now the second case, also we are putting the same red blood cell sample in each one. In the first one, we add anti-A and the second one anti-B. If we observe the first one, there is no agglutination, similar to the second one. Once we say there is no agglutination to exist in the two, mean there is no agglutinogen to be present on the surface of red blood cell. So, no agglutinogen to exist on the red blood cell, mean the blood group is O. At the end, I want to thank you, dear students, for your attention, and goodbye.